and welcome to my new YouTube channel Knowledge of Biology. Today I am discuss with you transcription of eukaryotes. Look at that. Transcription of So this transcription of eukaryote, this procedure occurs by this is our cytoplasm and then this is a small parts of nucleus. So in this nucleus are occurred by transcription. We know that nucleus are present our DNA. DNA are not capable to be directly producing proteins, so they are converted into the single stranded of mRNA. So this procedure we know that transcription. Okay, so into the nucleus, firstly DNA are converted to be mRNA, double stranded are converted to be single stranded. Okay, but this mRNA we know that as a pre-mRNA. Why this is called as pre-mRNA? Because this is unmature. This is not completely synthesis. Okay, so occupy into the three stages of this mRNA. First, capping. Second is splicing, and third is polyadenylase. Firstly, splicing. Into the splicing, the single stranded of mRNA have two portions, coding and non-coding part. For example, this blue line is a non-coding part of mRNA. This is known as intron, and this black portion is known as exon. It's a coding part, and this is a non-coding part of mRNA. Okay, so into the splicing process, applied enzyme splicosome. This splicosome enzyme having 18 proteins with 5 subunits, which is then U1, U2, U4, U5 and U6. These 5 as a cofactors responsible for this is the intron part of single stranded mRNA are removed. Okay, taken by screenshot. Next is first stage is applied as enzymes splicosome for the splicing procedure. So in this procedure, for example, it's our intron part. Okay, this intron part are attached to be 5 subunits U1, U2, U4, U5 and U6. They are pulled up to be this part of the intron and remove it. So we have just like a fragments. So these fragments also remove it splicosome as an enzyme. Okay, so then finally we get it axon, axon. Okay, so this is a complete for the splicing process. Next stage is, second is capping. After splicing, capping into the starting portion of single stranded mRNA have to be attached nucleotide with 2-phosphate, second nucleotide with single phosphate, and then nucleotides are reacted with guanyl transferase enzyme, means GTP converted to be GMP. Triphosphate are converted to be monophosphate. So these two phosphate groups are removed, have to be single phosphate group. NP and GP. Okay. Then reacted methyl transferase as enzyme to be responsible of attach methyl group up to be this starting portion of the mRNA. Okay. So we have GP. M7GP. Okay, and it's a axon. Take by screenshot. Then next procedure is a polyadenylate. So our mRNA, so axon, coding parts of this, and this is a M7GP. Okay, and into the 3 and portion of this segment we are applied to be polyadenylate. So we want to be attached nucleotides just like a. Okay, so this nucleotide attached responsible of the enzyme polyadenylase phosphatase. But one thing important to be this phosphatase enzyme are startingly into this process are inactive. Very important to be they are converted into the active form. So reacted as enzyme or the active form is a polyadenylase adenylate. Okay, so this enzyme phosphates are activated. Inactive converted to be active, and then we get it nucleotides. Okay, and it's axon portion, and it is okay. So this is a capping, starting portion of segment, and this is a splicing, and then end portion we are applied to be polyadenylase. This is have a complete mature mRNA. Take by screenshot. Very important to be mRNA are mature because it's a cytoplasm, it's a nucleus, and this present as a our DNA. Okay, and this is also occurred by transcription process. So this single non-mature, okay, mRNA try to be cross this nucleus pores, but this portion are present to be some proteins not allow it to be, they are passed out from this cytoplasm. because unmature mRNA are passed to be this cytoplasm, so this portion are present by hydrolytic